In this video, we will discuss about some of the bioinformatics analysis of the dysregulated genes, pathways, dysregulated genes, and the pathways that are related to these genes in the cell migration, differentiation, and proliferation assays that we have discussed in our previous videos. That how they did these three studies. So, first they find out the genes that are upregulated and the genes that are downregulated, and they run this sets of genes in the GSEA analysis to find out the pathways that are related to those upregulated and downregulated genes. And they can see that most of the pathways that are upregulated are related to this mitochondria translation, electron transport chains, mTOR signalings, and this homeopathic differentiations. Whereas the pathways that are downregulated is related to this transcription, mRNA processing, autophagy of mitochondria, and this FR1 signalings, and many other. Thereafter, they wanted to find out the relationship between the proliferations, migrations with respect to differentiations. So what I did is they made this plot where on the horizontal axis they plotted the volcano plots for this normalized volcano plot for the differentiation, and on the vertical axis they plotted for proliferations and migrations. And what they are able to find out is, for example, these mitochondrial translations genes are upregulated in the case of differentiations, but they are downregulated for proliferations and migrations, as it is on the negative side for proliferations and migrations. Then the genes such as the mTOR signaling, mTOR genes is been upregulated for differentiations but is downregulated in the proliferations and cell migrations. And the genes that is not expected is the ATIC genes. ATIC seems to be upregulated, highly upregulated in the case of differentiations, but it is highly downregulated for the proliferations and also for this migrations. And when they check the cells phenotype for these ATIC genes, ATIC knockdown, they can see that there is a change in the structures as the polarity goes down in the case where ATIC gene is been knocked down, compared to the control case where we can see the shape is more uniform. Then other things that surprise the researchers is about this mTOR signaling pathways, because with mTOR signaling pathways is related to the growth and proliferations of the cells. However, along with the mTOR pathways, they find the many other pathways that plays a role towards this mTOR1 signalings, that is shown in this figure, and the color dictates the enrichment, and the depletions is dictates by this orange color. So here it has been shown here that in this the schematic has been shown here, in which case this when the TCS1 is been knocked down, then this mTOR2 functionings will not occur. And however, when this RHEB for the mTOR1 functioning to occurs, they are related to this regulators that is been represented by LAM TOR 1, 2, 3, 4 and L5. These plays a role. These also plays a role towards this mTOR signaling pathways. But this P13K signaling pathways is usually activated by P13 PIK3CD and mTOR2, but which has been suppressed by this TSC1, which is upregulated in this case, which is upregulated in this differentiated neutrophils case. So, and one more thing that they find out is that most of the differentiated cells belongs to the neutrophils. So this is in brief some of the bioinformatics analysis, preliminary bioinformatics analysis that they have presented here in the case of the cell migration, differentiations, and proliferations because of the knockdown libraries that is been used in this study. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.